So welcome to the Data Core workshop. I'm Iris Lang. I'm the Data Core co-leader for ADRC at Wake Forest. For those who are not familiar with the Data Core Steering Committee, I'd like to give you an introduction of the committee. Let me see. So I'll be the first one to use this. That works. So the committee has 3 voting members. So Sheng Luo from Duke UNC ADRC is our current chair. Sheng, are you here? He's probably still flying because it's hard to, oh, there he is. He is our current chair. He'll be chairing the Data Core session later. And Erin Abner is our wonderful past chair. She cannot make it today, but are you online, Erin? I think she has a study section, but she's our past chair and I am the chair-elect. So officially, after the fall meeting, I'll be the chair. So as you can see, so we rotate out every 3 years, but every year we'll need a new member-elect. So look out for those emails if you are interested. But to be eligible, you have to be either the Data Core leader or Data Core co-leader, or that's the only eligibility criteria. And so we also have Nina Silverberg and Walter Kukul sitting on the committee. We meet monthly, usually less than 45 minutes. The main task of this steering committee is to organize this Data Core workshop and the Data Core session in 2 days. We also interact with NAC from time to time to talk about the Data Core related activities. Let's see. So congratulations to Anna Capuano from Rush ADRC as our member-elect for, so she will be the chair-elect after this meeting. And this year, we also initiate the Early Career Representative Program. And we have Pen Zhang from Vanderbilt EADRC and Raylin Liu from WashU Knight ADRC as our first cohort. Pen, are you here? There. And Raylin, oh, Raylin is standing there. She just came in. So their term is 2 years. They attend our monthly meeting as well, so they can see how we operate. And their task is to organize one event during their tenure, and then mainly target to the junior investigators. So look out for this email as well, okay? So talk to any of us if you have any questions, you're interested, any suggestions for the steering committee. And there we also have a networking opportunity. So I Googled up this restaurant. It's in the mid-range price. Will be 6 minutes walking from here. If you're interested, we'll meet at the lobby at 6. Honestly, I don't know where the lobby is because I came in from another door. But if you don't see anybody, just walk to the restaurant. Rain or shine, I will be there. So, today, right? It's today. So the meeting for the workshop will ending around 5. So I give you an hour to get yourself ready. And remember to bring a coat. I was told it's pretty cold in the night.
And Iris, I would just recommend everyone meet just in this hallway outside the ballroom here. That'll be easier.
See, another evidence. I need Hannah. Yes, let's just meet here at 6. So we all know here. Without further ado, we'll welcome our first speaker today, Dr. Yi Zhao. Dr. Yi Zhao is Associate Professor Su Hui Scholar in Biostatistics in the Department of Biostatistics and the Health Science Data Science at Indiana School of Medicine. She is the Indiana University ADRC Data Core co-leader. Welcome. And she will tell us about multimodal data integration. So here's it.
Thank you for the introduction. First, I want to thank the committee for offering me this opportunity to do the presentation. I just started serving as a co-leader since this fall, so this is so I'm really happy to join this big family. I'm going to talk about multi-view data integration. Oops. Oh, great. Okay, so what I'm trying to do is, so using some mediation or path analysis idea to try to integrate data collected from different modalities, and then also try to study the underlying biological mechanism. So this is our biological hypothesis. For example, we will start from the genome information, genotype, and then that will have an impact on the expression, and then also later the observation of the protein expression level, and then later as a phenotype, neuroimaging, and then cognition. So that's in the downstream of this whole path analysis. So under this directional assumption, then we probably can integrate data collected from different dimension or different modality using some like a path analysis or even mediation framework. So using an example in Alzheimer's disease, so for example, if we want to integrate some proteomics data with imaging data and then also cognition, so we have the assumption is that the CSF protein will have the impact of the brain volume or the level of brain atrophy, and then later have the impact on the cognition. So what we are trying to do is instead of looking at a specific marker, for example, A beta or tau, a specific brain region, but we want to start from the whole, all the protein we can measure, and then also the whole brain level, and then to try to identify if there's any new potential causal pathway. So then we wanted to integrate proteomics data. For example, we consider 320 peptides, and then also the mediator will be the brain volume from, for example, 145 brain regions, and then the outcome will be the cognition outcome, for example, the ADNI-MAM outcome in ADNI study. So right now, the challenge is that we have high-dimensional exposure, high-dimensional mediator, and then we also have like some the dependence within the exposure and the mediator. So how can we articulate the relationship? So just a brief introduction about mediation if you're not familiar with. So for mediation analysis, we try to articulate or decompose the treatment effect on the exposure into the part through a third variable called mediator, and also the part not through the mediator. So it helps quantify the intermediate effect and then also clarify the underlying causal mechanism. A pretty traditional way of doing mediation analysis is using the structural equation modeling under this linear assumption or parametric assumption. So we have the mediator model and then also the outcome model. And then we, under some certain assumptions, so we can show the alpha beta product will be the mediation effect, and then this gamma parameter will be the indirect effect. So now what we have, so just introduce some notations. So now we have the high-dimensional exposure case and a high-dimensional mediator case and a scalar outcome. So then I use this xi and then mi to represent those matrix to represent the exposure and the mediator. And then the question is now we have dependent exposures. So for those peptide or intensity, so we will, for example, the peptides from the same protein, so their intensity level should be dependent. So then how can we, if we ignore this dependence in the exposure, so we may have some bias estimation in the downstream causal effects. So then what we did is like consider this pretty straightforward and a simple way just to do a PCA to just to remove the dependencies and then generating those independent exposure components, and then we can just continue our analysis. So suppose right now we have the independent exposure components, and then later I will talk about how we can interpret those components. And then we can draw a causal diagram. So we have those independent exposures, and then the dependent mediators, and then an outcome. So the next question is how can we get around the dependence between the mediators? Because if we consider the brain regions as the mediators, even though they are dependent, we do not know the causal dependencies. So then it is harder to just, if we ignore the dependence, so we'll get biased results, but we don't know how to just have the causal sequence of the mediator. So then we consider a marginal model to try to address. So basically, we just consider the marginal effects of the mediator to account for the dependency of the model, the dependencies of the mediator, and then we summarize those dependencies into the model error terms. So we all know in regression, so if the model error terms are dependent, if we use OLS, we still get unbiased estimation or consistent estimation of the model coefficients. So that's what we did. So we just consider the marginal effects. So, and then in our paper, we also show that if we just enter this marginal formulation, so the interpretation of the mediation effect is the mediator-induced mediation effect, basically meaning if this mediator is in the causal sequence next, closest to the outcome or the last mediator in the causal sequence, then that's the interpretation of the mediation effect or the parametrization under this marginal model. So this will also give us a pretty informative information about the mediator. That's what we call the mediator-induced mediation effect. And then the next question is how can we do the estimation? So we have the high dimensional parameters. So then we just use some like a regularization. So what we consider, we have 3 different parts of the regularization. So the first part will be the what we call the pathway lasso penalty. So basically we group we directly penalize on those mediation effect. That's the product of alpha beta, and then we try to achieve the goal of mediation selection. And then the second part is a group lasso penalty. So basically we group those mediation effects from the same exposure. Then this will achieve the exposure selection purpose. And then lastly, so for the third part, we will just penalize the direct effect. So this is the 3 parts. And then so we have the regularization, and then also just we can use some augmented Lagrangian, and then to estimate the parameters, and then we also use some like a BIC to the tuning parameter section, which is pretty general or traditional in the regularization regression. Just to go to the ADNI application, so we have 135 MCI patients. We look at the exposure as the CS or proteomics, and then the mediator is the brain volume data, and then the outcome is ADNI-MAM cognition outcome. And then we also did some confounding adjustment for age, gender, and then here we also consider genetic marker ApoE4 and then years of education. Sorry. Okay, so this is our result. So first we use PCA to remove the dependency in the exposure. So we just consider the first 20 PCs, which already account for over 85% of data variation. And then when we do regularization, so on these, and then the 145 mediators, so these are the causal pathway we identify. So I will go into those more details. So those are the PCs, and then the corresponding estimation of the mediation effect. So we will see some strong effects from PC1, PC4, and then PC5. And then if we look at those PCs, and then these are the proteins, and then so these proteins we identified is also identified in the existing literature, and then also we look at the brain mediator. So here we want to highlight, so we have, even though all the indirect effect is positive, however, the mechanism will be different. So for the cortical or subcortical regions, so the mechanism is the increase of the protein level will, in the corresponding component, will have a decrease in the brain volume, and then that will decrease the cognition, sorry, a decrease in the volume, that will decrease the cognition behavior. However, in the lateral ventricle region, so then we have the opposite mechanism. So this is also consistent with our knowledge about our brain and an AD. And then this is just a summary of those key regions. We identified those are, most of them are AD markers already identified in the existing literature. And then we use this figure to summarize our results. So we identify some like a protein PCs, and then those protein together will have an impact on the brain volume, and then later have an impact on the cognition behavior. So this is a way to do multivariate integration using some mediation analysis. So we just can account for the case with high dimensional exposure and a mediator. However, one criticized on this approach is that we cannot do inference. So then later we considered another approach. So we simplify the problem a little bit. So suppose we can identify an exposure component, and then we can also identify a corresponding mediator component, and then we will have parallel mediation pathways. And then we can either specify the loading profile of those components, and then to do an interpretation. Sorry, I didn't mention here. So when we look at those CSF protein components, we just use some like a lasso approach to specify those loading profile, like sparse PCA. So this is another way. So then the goal is to identify those components, those phi and then psi parameters, and then also the model coefficient at the same time. And then this will also offer an opportunity to do some inference. So then if we apply to the same data set, so we actually identify pretty consistent results. So those are the proteins and then the brain volume. So right now you see the arrows have color because now we have the component one-to-one correspondence. Then we can also draw the inference and then estimate the sign of the corresponding to each component. Another approach we did for data integration using mediation idea is to, so suppose we have two high dimensional mediator blocks in the middle. So for example, if you want to have some like a scalar exposure or genetic exposure, and then try to look at the connection between two imaging modality and then the outcome. So for example, we have an exposure, and then we have the structural connectivity and then the functional connectivity. The biological assumption is that the structural connectivity may inform the functional connectivity, and then we can integrate those. And then still, we don't want to have those two modality as a single big block of mediator because we want to separate those and then see if there's any connection between the two. So this is the idea we had earlier, just to do a mediation with two blocks of high dimensional mediator. Again, so when we apply to, this is now Alzheimer's disease analysis, but when we apply to the human connection project, so we wanted to study how can brain explain the sex difference in the cognition behavior. Then we identify the corresponding DTI and the functional MRI networks. So, and then if we do the interpretation, so we can just characterize those regions into two, so the working memory and then language. So for a language behavior, this picture vocabulary, which also related to some working memory, so it also explains how the sex difference is not only just because of the difference in the language, but also related to working memory and then the strategy of memory retrieval, basically. So, yeah, I think this is pretty much what I have, and then just give you some idea of how can we do data integration using a more hypothesis-driven approach, like considering some path analysis and mediation analysis. And then I want to thank Annie and HGP making the data publicly available, and then my funding resource. And then thank you so much for your attention.
We have about 3 minutes for questions.
Hi, very nice talk. It's nice to see all this mediation analysis, especially through the perspective of high-dimensional data. The first question is, Was your data standardized? How did you take into account the variability from all these blood markers that were collected differently or from the brain imaging data?
So what I did right now, so they are all from the same study. So like the proteomics data is from the ADNI database. I haven't thought about like a data harmonization type of question. Yeah, but that could be a good point. So, but for the HCP data, I just use HCP like a minimally pre-processed data.
Did you look at the variability from across centers? How?
No, not really. So, but yeah, that's a good point.
Thank you.
Thank you.
I think that's a very cool approach. Next, we have Dr. Hui Zhang. He is the director of Northwestern ADRC, and he's a professor of preventive medicine in the division of biostatistics. Welcome. And he will tell us about co-localization in Alzheimer's disease research images.
Good afternoon, everyone. And I'm from Northwestern University at Chicago. And at Alzheimer's disease, we know a very important protein called the amyloid beta play a very important role in the Alzheimer's disease. This is a Nature communication paper a couple of years ago, tell the interaction between another protein called the TDP43 versus the amyloid beta. But you can see this image here. Okay, you can see on this image, on the upper left corner, that's the green color, means the amyloid beta. And the TDP43, the image, the red signal below. But how to see their interactions? Biologists, how do they study the interactions? So they want to show, okay, if these two proteins under the microscope is together, they say, okay, maybe these two proteins have interactions. That's the best way they can do. They can prove. You cannot see the direct interactions. So the way they show the interactions is something called co-localization of these two different protein molecules. That's the right lower corner here with the yellow color. When they see the yellow color, they say, okay, the red one and the green one are together, are co-localized. They think, okay, these two proteins have interactions. So that's a very important image for the Alzheimer's disease research. However, biologists understand this picture very well, but as a statistician or data scientist, we don't know why. Why one protein shows green color, why another protein shows red color, why together they show yellow color. So let's review something. This is because they use a light microscope. These are light signals. But let's review what's light in high school we learned. So in high school we learned the light actually has two physical properties. Number one, light is a wave. Number two, light is a particle. So as a wave, actually the wave covers long range. But whatever the wavelength can be detected by our human eyes, by our retina, we call the visible light or visible wave. This covers the wavelength probably from 400 nanometer to 750 nanometer. That's a visible light human can detect, can see. So as a wave, it has wavelengths, right? They have wavelengths. As a particle, it has energy. So we learn in the high school physics, when the wavelength is shorter, the energy is higher. That's why the UV is more dangerous. That's why the more on the right side is more safer. So as a particle, it has energy. So that's why the proton protein shows the green color. That's a Nobel Prize in 2008. So what they did is, okay, they found some proteins in the sea animals, under the sea. This protein they found these natural proteins. When you give a laser with a blue light laser, and this protein will absorb some energy, and then release the rest energy. So because they absorb some energy, the energy they release is less. Less energy means the higher wavelengths. So they will release the green color. So that's why if you take this protein to a target protein under the microscope, you will see your target protein with the green color. Because when you use the blue laser to excite it, they will release the green color. So this way you see the green protein under the microscope. And the biologists modify this protein and to another protein called the red protein. If you also give a laser, the blue laser, it will release the red color. That's how you can see the one protein, the amyloid beta, you can see the green color. The TDP43, you can see the red color. But when they are together, what happened is like this. Let's go back to this slide. So when they are together, if you mix the green color and the red color, you will see the color in the middle, which is the yellow color. That's how could they see, they label one protein with red, another protein with green. And if they are together, we call the co-localization, you will see the yellow color. But the yellow color, they can tell, okay, these two proteins have interactions and the current experimental status. This is how it works. But remember, this is from the light microscope. As I mentioned a moment ago, light is a wave. From physics, we know, okay, as a wave, it has some basic physics properties. The wave has some property called diffraction. Okay, it means that, okay, if you have the light go through a small aperture, it will actually, they will go to the more wider area. It's called a diffraction. If you have a laser go through a very small point, after that point, you will see some ring outside the ring. That's because diffraction. So this is because this diffraction, if you use the light microscope, the best resolution, if you have all the conditions perfect, ideal, the best resolution you get is around like maybe 400 nanometers. You cannot get the resolution better than this because the light has the diffraction. But how to further improve the resolution of microscope? That's a Nobel Prize in 2014. So they have a technique called the SMLM, or single molecular localization microscope. This is a complex technology. But think about a simple way. We are statisticians, we know what's normal distribution, right? For normal distribution, like a bell shape, if you cut off two tails on the left side, the right side, it's called the truncated normal. You only keep the signal in the middle, you will remove some noise, right? But the similar idea, we can further improve the resolution of the light microscope to 20 to 30 nanometer. So you can see from this image, I don't have a mouse. Okay, you can see the image on the right lower corner. You can see before this technology and after this technology, this is for the cell skeleton. You can see the resolution improved a lot. But now, some trouble happened for what we have introduced a moment ago. For example, on the right up corner, remember some protein is the red signal, some protein is the green signal. The way you tell the localization, you see the yellow signal. But now, if you have much better resolution, like a right lower corner, the resolution is much higher. You won't be able to see the yellow anymore because the resolution is so good, right? And also another way, sometimes for protein A, protein B, sometimes they like to be together. Sometimes they don't like to be together. They're not random. They try not to be far away from each other. The yellow color can only tell you if co-localized or not, but they cannot tell you the reverse conclusion. They try to repel from each other. So now it gives us a challenge, a statistical question. So how to quantify the co-localization and for the new microscope image with high resolution? So previously you have a binary answer, yellow color or not, it's 0 or 1, right? But actually, for this case, biology is like an answer from a -1 to 1, like a Pearson correlation. 0 means, okay, the two proteins have no relationship, both are randomly, they have no relationship. Further higher, like a 0.9 or 0.8, I mean, they are very likely to be collided together. The negative number, they don't like to be together, like a -0.8 or -0.9. So they need this kind of index. But we studied in literature, we didn't see any this kind of index. We saw a method called the CBC method, but it doesn't work very well. I will compare our method with them. That's why we proposed a new method to propose a new statistical method to quantify the correlation here. And this paper was published 2 years ago on the SMMR. The further author was my postdoc. Now, this year she rather got tenure from New Orleans. So in this paper, we can see the up image is a real data image. And the resolution is so high, they are giving us the 2D coordinates or 3D coordinates of each single protein molecules. But these coordinates, we replot these coordinates to an image, that's an image below. We try to quantify the correlation between the red and the green signals. But the question is, why couldn't we use something like a Pearson correlation? That's very natural, right? Why couldn't we use it? We have two challenges. So if we simulate like here, like this image, if our signal is a completely spatial randomness, so the red and green have no relationship. But if you use Pearson correlation, if your signal density is high enough, for example, you have 1,000 red signals, you have 1,000 green signals. If your cell density is high enough, even both of them completely spatial randomness, the Pearson correlation will be very close to 1, will increase density. That's why the bias estimation. Another challenge is how about the signal on the edge, on the boundary. For example, for some red signal on the boundary, on the edge, they only have chance to have correlation on the green signal on one side, but another side they don't have chance because it's the boundary of your microscope image. So that's unfair for the some signal on the boundary. That's why to address these two challenges, we propose a new method. We call the we try to propose a new method to address these two challenges. One is the edge effect correction. Another, we try to normalize this index to the range from -1 to 1. So I will try to skip the technical details. If you are interested on the technical details, you can go to our paper because we only have 15 minutes. So we use two types of normalization. One is Z type, one is D type. One is use like a Gaussian distribution, one is use the ratio. So we define this index called normalized spatial intensity correlation. And this is a pretty complex definition, but we successfully address the two challenges. And I will skip this very quickly. Another question is how to choose the range. For example, if you say, okay, these two protein molecules are together or co-localized, but how close they are, you will claim a correlation. For example, if I claim, okay, somebody is with me this afternoon. So when you define the distance, right? If this distance is like 50 miles, so everybody in Boston is with me this afternoon. If this distance is like 3 feet, nobody is together with me this afternoon, right? So you have to define this neighborhood, the distance. But how to define distance? Many times we rely on our collaborators. But sometimes collaborators don't have a good answer. They say, okay, by your common sense. But we don't know, right? Sometimes we also need to calculate this index among a range, like use the calculus, and then take the average across this range. Sometimes we use some like a very common sense for some biological questions. So that's why we propose a method to calculate the distance, like at the bottom here, use the calculus to calculate the distance among range and take the average. And we try to compare our method with the only available method called the CBC method. This method can only compare the correlation of 2 signals, not 3 signals. And sometimes we need to do that. And here's the simulation. You can see the left up corner is the similar data we generated. On the first one is the red and green signals. They have very good overlap, but of course with some noise. And then the first one is called the complete spatial randomness. So for the third one is like they are not together. They try not to be together. The second one is very complex. The partially overlap and partially not to be together. And you can see our method. I have to come here. I think I need to show you. Well, this is some simulation to show our method is robust and reliable compared with the CBC method. And I will skip this one. Maybe this go to the next one. So you can see this one is also simulation for another different case. And for the third one, you can see these 2 signals. The red and green have very good correlation. The first 2 are complex, like cross. You know, in the center, they collide together. On the edge, on the surrounding area, they are not to be together. And the first one is more complex. We have 3 colors, 3 different signals. Our method, you know, is reliable for all the cases. And the CBC, most time, you know, they mess up. And for the last case, for 3 colors, they cannot do it. And maybe I will skip this and given our time limit. So we apply our method to real data. This is real data in the immunology research. Another real data is for the Alzheimer's disease research. So this is some signals in the hippocampus or the red. You can see the hippocampus red. By this method, we successfully detect the correlation of the two signals, the TDP43 and the amyloid beta in the hippocampus of the red. And because this definition, this index is quite complex, so we provide this R package. We check the download for this R package. We notice that this R package have more than 20,000 downloads. This is not a very popular question, like a ggplot2, probably they have millions of downloads. But this one is not too many people are interested on this question. We think this is a good number of downloads. Now we also checked who is downloaded, from which country. We checked for only 1 month, 30 days. We found that the user actually from all the world, not only from the research active countries like China, United States, like England, Japan. It's actually everywhere. In many places like Ukraine and Vietnam, lots of people still use it. And because the R package would need a coding ability, lots of biologists they don't know how to code. So we provide this online Shine EPP. So you can just go to the online Shine EPP, click a couple of buttons, you can calculate this index. And for lots of people, they say, okay, they are putting the patient data. They cannot go to the external server. They have to keep the data on the local computers within universities. That's why we also provide this desktop software. You can download like a software and install on your own computer. So, and this method we have used for some collaborators. For example, we have published a multiple paper. This is a Nature paper. This is Nature Immunology. Another is a molecular cell. Another Nature Immunology. Since I think this is most of last year published. And so what's next? How much time do I have? Okay, give me maybe 2 or 3 minutes. So we introduced, the new microscope a moment ago. But now, think about this. When you use the blue laser to the protein, and let the protein release the green lights, the blue light has a shorter wavelength, have more damage on your cell tissue, on your proteins. That's why sometimes your cell, your tissue will die very quickly. But now we have the biologists have a new microscope called a 2-photon microscope. Previously we used the blue laser, but now they use the red laser. Remember, red light has a longer wavelength. It's more soft, more gentle. And your protein will absorb the 2 photons, red photons, rather than a single blue photons. And they still release the green colors. But this way, you can keep your tissue alive because it's much softer, much gentle with the red lights. By this way, we can track the real-time activity of your cells. And for example, for your neurons. For neurons, one neuron talk to another neuron, they use some structure called a synapse. One neuron release some chemical particles will captured by the second neuron. They will call the action potential. That's how the signal transduction in our brain. So this is some real data from Stanford University, my friend lab. So you can see this is neurons dendrites. On the right lower corner is a cell body. It is like a center of the neuron. And this is white long line is like called dendrites. On the dendrites, you can have lots of like a small white spots. White spot is potential space to have the signal spike, or called the action potentials. You can see some small red circles. I mean, they recorded, using this new microscope, they recorded the active signal for these small circles. But for them, the question is, okay, for these active signals, they count the distance for this signal from this location to the cell body, to the center. That's a one-dimensional data. They only count the distance. They want to see, are these signals clustered together or well spread? That's a correlation of the single signal to itself as the one-dimensional data. They think they answer this question will help them to understand the learn and the memory in our brain. They think that this is a cellular mechanism for the learn and memory, but they don't know how to deal with this kind of data. This is the correlation of a single signal with the one dimension. This is one example. Another example, I will skip this. So you can see this is a real-time movie recorded at Stanford University. Don't know if I can play it. I need a mouse to play it. Okay, can you play this movie? Can you use the mouse? If you move mouse cursor, you will see a play cursor here. You cannot play. Oh, that's fine. I will use this one. Okay. So this is real movies recorded at Stanford University. So on the upper one is some control mouse. You can see on the right side it recorded signals for 12 different neurons along the time. You know, at different time they have the different signal spikes. The lower one is the Parkinson disease mouse model, similar like Alzheimer's disease. They record this. You can on the Parkinson mouse, these neurons try to have a spike, have activation together simultaneously. That's why you can see the Parkinson disease patient, they have their hands shaking because all the neurons are active together. Their hands shaking. So for this data, what they are interested in is, okay, along the time, this is one dimension, right? Along the time, are these signals that occur simultaneously or not? So in another language is a co-localization of your signal along the time, along the one dimension, time. So how to analyze this kind of data? Currently, they use a method called the replicate function. But I don't think that's a very good method. We are trying to develop a new method for this one. But it takes our method to the one-dimension data, to a single signal, single type signal. Okay, I think that's all. Thank you, and I'm ready to take some questions.
Unfortunately, we are out of time for questions for this speaker, but we do have a 25-minute break coming up, so maybe you can find them during the break. Thank you.
Well, thank you, Dr. Zhang, for the education on the high school light physics and a very cutting-edge technique as well. We're looking forward to your next adventure on the colocalization in the time. Yep. All right, next speaker will be Richu Stukwish. I'm sorry if I pronounce your last name wrong. Richu is a grad student researcher at UAB ADRC Data Core, primarily working as a liaison between the OR core and the data core. Welcome, Richu. And her talk is on the joint modeling on the cognitive trajectories.
Okay, hi everybody. As I said, I'm Rachel and I'm a DRPH candidate at UAB in the Department of Biostats. So I'm going to talk about the applications of joint models for assessing attrition bias when studying cognitive trajectories. Okay, so first I'm just going to review the summary of I'm just going to be reviewing missing data concepts for everybody, and then I'll introduce the joint model, followed by a practical example using cognitive data from the REGARD study at UAB. So as most researchers learn, missing data is going to fall under 3 categories: missing completely at random, missing at random, and missing not at random. Correctly defining a study's missingness assumptions impacts your statistical modeling choices used during analysis, and therefore minimizing potential bias related to study attrition. When our data is assumed to be missing completely at random, both linear mixed models and models using generalized estimating equations are applicable. This assumption remains unbiased and can kind of be thought of like that gold standard aim hope that you can have. But when data is assumed to be missing at random, only linear mixed models are applicable. Now, neither linear mixed models nor models using generalized estimating equations are valid under the missing not at random assumption. Unfortunately, we cannot tell from the data on hand whether the missingness assumption is considered missing at random or missing not at random. So when does missing not at random occur? Often missing not at random occurs when studying competing risks. And what can we do to address data that is potentially missing not at random? Nope, did I go? Okay. So joint models for longitudinal and time to event data fall under this umbrella term of shared parameter models. They're created via a modeling method that considers the dependency and association between longitudinal data and time to event data. That association between the longitudinal process and the time to event process is explained by their shared random effects. So when might we need these joint models? So joint models can be used in a survival analysis context to handle endogenous time-varying covariates, but they're also used when studying longitudinal outcomes, provided that the joint model is well specified to account for non-random dropout. So while various other models exist for handling data that is missing not at random, a unique property of the joint model is that when the longitudinal outcomes are of a linear combination and occur in the presence of dropout and death, the estimates provided by the subject-specific models are interpretable as subject-specific effects in the population alive, whereas marginal models estimated by generalized estimating equations provide estimates of the population averaged effects in the population currently observed, or in the population currently alive, respectively. So, in simpler terms, joint models are able to provide the same benefits of a linear mixed model inference while getting potentially less biased estimates. So here we have the basic structure of a joint longitudinal survival model. The linear mixed model must be built first, as this function is where we obtain the trajectory function. After determining the trajectory function, we are able to build out the time to event or survival model. So typically in an ordinary Cox regression, the baseline hazard function is left unspecified. However, if an unspecified hazard is applied in a joint model, it's been shown that the standard errors in the parameter estimates can be underestimated. To avoid this issue, parametric but flexible functions are often recommended to use for the baseline hazard function, such as like a piecewise, constant, or spline models. One of the most important items of a survival submodel is its association parameter. The association parameter measures the strength of the association between the longitudinal process and the survival process. So, on to a practical example. We know the negative effects of smoking on physical health and mortality are widely consistent, but studies focusing on the effect of smoking on cognitive impairment in aging populations have historically presented conflicting reports. A repeated concern in the previous studies is the lack of consideration for the effect of differing mortality rates between non-smokers and smokers, and the impact survival bias may have on results. What might be that cause? The competing risks. Because smoking is known to increase an individual's risk of death or dropout in an aging population, the longitudinal cognitive follow-up among smokers may be truncated, leaving only a healthy population for analysis in later ages. Therefore, the goal of this study is to estimate the effect of smoking status on cognitive trajectories in aging populations while accounting for potential bias due to attrition. 2 longitudinal models will be used, a linear mixed model and a joint survival model. So, for those who don't know, REGARDS is an epidemiological study following a cohort of US adults for stroke and cognitive decline. Participant recruitment occurred from 2003 to 2007, and more than 30,000 US residents 45 years or older were recruited. The cohort includes only African Americans and European Americans, and approximately 1/2 are European American and 1/2 are African American. While participant contact occurs every 6 months, The regards enhanced cognitive battery is conducted during follow-up calls at 2-year intervals. And their enhanced cognitive battery assessments include the 6-item screener, wordless learning, delayed recall, the letter F, animal naming, and a modified MoCA. So I'm sure some of you are wondering why I'm showing an example using regards at a NAC conference, and how is this applicable here? So the simple version is that the regards enhanced cognitive battery can be viewed as a subset of the NAC C2 neuropsychological battery. Specifically, each assessment performed within the regards enhanced cognitive battery is also given in the NAC C2. So key differences include the use of MoCA in the overall cognition categories. So NAC includes the full MoCA, but regards administers a modified version of MoCA known as the 5-minute battery. This mini MoCA was developed as an alternative, and it's to the mini mental state examination and designed to detect individuals with mild cognitive impairment. So the modified 5-minute battery in regards includes only memory and orientation items. Additionally, the regards assessments are categorized as memory and verbal fluency, and NAC includes an additional category of attention. Finally, NAC assessments are performed annually and in person, while regards cognitive follow-ups are performed every 2 years and over the telephone. Both regards and NAC have creative cognitive normative values. In regards, cognitive Z-scores based on sex, age group, normative groups that were calculated using the means and standard deviations for each assessment by sex and age groups. Each time point has a composite Z-score, and each available Z-scores were averaged to create the following composite groups: memory, fluency, and overall. There we go. Okay. So our primary outcome was the composite cognitive score for each participant. The exposure was the participant-reported smoking status, categorized as current, past, or never. The covariates are defined as your practice effect, which is a time-dependent variable designating if the score was the first assessment or not. Participants at baseline were centered at 65 years of age, or the participant age was centered at baseline at 65 years. Their sex was male or female, and race was Black, African American, or white. Education level was high school graduate or less, or college education or above. And then we also included self-reported alcohol use. The random effects included were intercept, time, and time squared. Time was defined as the follow-up time since entry into the study and measured in years. To account for the curvature present in cognitive trajectories, the models were fit with fixed and the random linear and quadratic terms for time. And that inclusion of that squared slope is consistent with previous longitudinal analyses performed for regards. The linear mixed model, our model testing, included comparing linear mixed models with random linear, quadratic, and cubic B-splines, and the final model the final joint model retained a dependent structure based on the current value and slope, and that was determined based on likelihood ratio tests. So now the results for overall composite cognitive scores. Current smokers showed lower average estimated composite cognitive scores over time compared to past and never smokers. As anticipated, across the 3 smoking status groups, the average estimated composite scores remained equal across statistical models until about 4 years in the study, where the joint models suggest a mild increase in the rate of decline relative to the linear mixed model. This is because at baseline, when the estimated cognitive differences in trajectories between smoking statuses are comparable in the linear mixed model and the joint model, every participant was still alive and observed. As duration in the study continues, The difference in the estimated cognitive scores between the statistical models within the respective smoking groups gradually increases. The image on the left shows our results for the memory composite cognitive scores, while the image on the right is for the verbal fluency composite cognitive scores. First, I'll talk about the memory outcome. Apparent all smoking status groups estimated memory composite cognitive scores initially rose before reaching a peak at approximately 8 to 10 years, then showed a decline. Overall, from baseline to 14 years in the study, there was an increase in the estimated difference of the mean memory score between current smokers and never smokers. In contrast, the memory domain analysis, the average composite scores in verbal fluency across smoking status groups remained relatively consistent over time, with a mild decrease occurring in current and past smokers. This is evident in the change in the estimated difference of the mean verbal fluency score comparing current smokers, never smokers from baseline to 14 years in the study. So what did completing the study tell us? The first is that the study provides evidence suggesting that increased rates of cognitive decline in smokers compared with non-smokers and former smokers, both in the linear mixed model and after accounting for differential attrition in the joint model. Spanning the three outcomes, current smokers had lower average composite cognitive scores in comparison to individuals who identified as never or past smokers, and these differences in composite cognitive scores slowly increased over time. The second is that across all composite cognitive outcomes, as time and study increased, the joint model predicted a marginally steeper decline in smokers versus non-smokers compared to the linear mixed model. Now, yes, while the linear mixed model and joint estimates are not drastically different in this example, it does show that joint models can and should be used as a sensitivity analysis at a minimum to ensure the impacts of potential attrition bias are evaluated. So we can say that we can see that as time in the study increased, the two models show a slowly widening difference in estimates, highlighting the different ways the models are accounting for death and dropout. And finally, within the analysis using NAC data, joint models can serve as a powerful tool for assessing and correcting for differential attrition, especially when the research involves competing risk for death and dropout. So that's it.
Thank you. We do have one question from the online audience. The question is, cognitive test scores are internal covariates for the death time. How do you make sense of it in the survival analysis?
Can you repeat that question? Sorry.
Yes, cognitive test scores are internal covariates for the death time. How do you make sense of it in the survival analysis?
So the way, if I'm understanding the question correctly, is that if we were to go back, go back to like the equation, you would see that the predicted, that trajectory function that has estimated their cognitive score is now becoming additional covariate to help to see if that score being high or low might be impacting, when their dropout or death occurred, which in the analysis, we were not designating a difference between death or dropout. It was just they were no longer showing up or had no future assessments provided. So, but we only designated the difference when we just did the survival analysis to show that they were related. But yeah, I hope that answers the question. I'm not sure if I understood it correctly though.
We have time for one more question.
Very interesting talk. Shen Lu from Duke UNC ADRC. So two questions. So one is that looks like you are using this joint model to handle this monotone missing, right? Due to death or dropout, meaning that after death or dropout, you don't observe that anymore, right? So have you thought about how to handle the non-monotone missing? Meaning that have some missed visit in the joint model framework?
So no, not really. We didn't think about the other missing data. It was just really in terms of that, the final outcome, because all the others were based off of a baseline initial measurement.
Okay. A second quick question is that looks like you have a multiple longitudinal outcome, right? So have you thought about how to analyze all these longitudinal outcome jointly with your survival?
So that's what this one does. So it puts them together into being a joint one. I'm assuming I'm kind of following what you're saying, is that the joint model does combine those together, but it was the way that they're now accounting for it. So once you apply that trajectory function into the survival one, ends up combining them, that's how you're getting them together to account for it. I think that answered.
Thank you.
Well, congratulations, Richu, and best wishes for your PhD study and research in ADRD. So next speaker will be online, will be Jingjing Zhou from UCSD ADRC. Jingjing is assistant professor of biostatistics, and she is going to tell us about the novel functional data analysis and machine learning for eye track in preclinical AD.
Hello, everyone. Want to make sure everyone in the room can hear me.
Yes, we can hear you.
Okay, that's good. Thank you. And thank you very much for organizing this workshop, and also a lot of thanks for the committee for selecting my talk and giving me this opportunity. So today I want to talk about an ongoing project at UCSD. First of all, as an introduction, I'm assistant professor in biostats at UCSD, also the co-leader of the data core at UCSD ADRC. So today I want to talk about an ongoing project on eye tracking, especially using functional data analysis and machine learning approaches for novel eye tracking markers in Alzheimer's disease. So first, just as a background, as is shown in this continuum graph below, there are multiple stages in Alzheimer's disease, especially in the preclinical AD stage. There usually are no symptoms, but possible biological changes in the brain, and then it progresses into mild cognitive impairment due to AD with very mild symptoms that may not interfere with everyday daily activities, and then it goes on to dementia, which is mild and then moderate and severe. So the AD begins, the pathological changes actually can be decades prior to the clinical onset of dementia. So that highlights the importance of early detection for managing this disease progression. And also early detection will be meaningful in terms of interventions, which are most effective at early stages. So there's an urgent need for low-cost methods for detecting and staging AD, especially from preclinical to symptomatic stages. And those methods are also very useful in identifying pathways for early interventions and also for preparing long-term care, because Alzheimer's disease is very costly in terms of the care. So currently there are multiple types of biomarkers that are used to for diagnosing Alzheimer's. For example, there are CSF markers, plasma markers, imaging, and genetics markers. But biomarkers crucial for AD detection often fail to correlate with cognitive functioning, especially in preclinical AD. So there is a difference in the diagnostic versus prognostic markers. And in particular, the biomarkers sometimes lack prognostic clarity on the timeline of the cognitive decline and clinical decline. And also, they can be expensive or invasive. For example, the imaging markers can be quite expensive to collect, which has a potential impact on the sample size that's available for researchers because of the cost. And also some markers can be invasive to collect. So there is a need for inexpensive and effective markers for early stage Alzheimer's disease. And that's why the novel digital markers, especially the eye tracking markers, come into the picture, in particular in certain tasks to detect and stage early stage AD. The scientific premise is in Alzheimer's disease, the early synaptic dysfunction can disrupt neural networks and which can particularly affect the task that require binding, so conjunctive binding, which is like the integration of various sensory inputs into a coherent representation. And I will explain what kind of binding task we are looking at in this study. And in this kind of tasks, eye tracking has been an emerging novel digital marker collected from digital devices like cameras, trackers, or even like wearables or mobile devices, because it is economical and also non-invasive. And there have been research on the utility of such eye tracking markers, including, for example, the eye gaze. That's where the participants are staring at during the tasks, or the pupillary response, like the size of the pupil during the tasks. And both modalities have been used in studies, and there have been papers showing their effectiveness in predictive of AD progression. And I'm listing several papers in here by this ever-growing research field. So here I want to talk about the research that's undergoing at UCSD. We are working with the data collected from 2 binding tasks sponsored by NIH. PI is Dr. Dan Jacobs. And in this study, we have 2 types of binding. The first one is so-called visual sensory binding, and the second one is a visual short-term memory binding. And in particular, in this presentation, I'm focusing on the second one, the visual short-term memory binding, which are basically a memory test. So the participants come in and then sit in front of the computer screen, and they are shown 2 sets of visual or memory stimuli, which are 2 or 3 shapes that can be different or same between the first set and the second set. And then they are asked whether this second set of shapes are the same or different from the first set. So the first set is called a study display or study stimuli. The second set is called recall. And there are 2 types of such tasks. One type only involves different shapes. So there's no different color involved. And then the second is the shape color task. We call it a binding task because it combines both shape and color. And either the shape or the color can be different from the first set of geometric shapes. For each participant, they are asked to do 32 trials, 16 shape only and 16 binding tasks. And during the task, their eye movements are captured by this Tobii Pro Fusion Eye Tracker, which is a research-grade eye tracker installed on the bottom of the screen. And it is calibrated, so it's pretty accurate. It captures both the pupil size and the eye gaze at high frequency of 250 Hertz. So it's a high frequency time series for both modality. And for the pupil size, it's just a 1-dimensional pupil size. And for the eye gaze, it's a 2-dimensional XY coordinate where the eyes are looking at on the screen. So this is a timeline for the short-term memory, a visual short-term memory binding task. And important stages are the second, third, and fourth squares, which indicates a 2 seconds of a study display where participants see these shapes on the screen for 2 seconds, and then 1 second of blank screen. And then another set of shapes appear either the same or different from the previous study display. And this stage doesn't have a time limit. So the participant can take their time to answer the question whether they are the same or different from before. And also, it can be 2 shapes or 3 shapes. So there's a parameter in this task, set size, either 2 or 3, which have different difficulty for the participants. So the overall goal of this statistical analysis is to use functional data analysis approaches, which is different from the widely used current approach, which relies on simplified single-dimensional summary statistics. For example, fixation duration, like how long the eyes are staring at the target, and saccade amplitude, so the magnitude where like the eye of moving from one fixation to another fixation. And we want to use the functional principal component analysis in particular in this presentation to understand the modes, especially like how the pupil size are changing, like how the trajectories are changing, and how different modes can represent the main modes of the pupil size trajectory among the participants in the cohort, as opposed to suppressing them into summary statistics. So hopefully to preserve more information and reveal more information that's related to the cognitive function and other biomarkers. And also, we are actually looking at different groups and hopefully to find interesting findings between the correlations between the groups and the pupil size trajectory patterns. So the groups we are looking at here is a 2x2 group given by the cognitive function, either MCI or cognitively unimpaired, so cognitive normal, and also across the biomarker p-tau 181. So we have a 2x2 group in this study. And we want to understand the variability in the eye tracking records. And in the future, because this is a longitudinal study, we actually have an opportunity to look at the longitudinal changes in the ET patterns and how they're related to the markers and cognitive function. So first of all, I want to show the result, just a summary of the accuracy, averaging all the trials for each subject, but looking separately for different groups. As I mentioned, cognitive function times p-tau, either positive or negative, and also stratifying on different parameters of the task. The binding means it contains both color and shape means it's only shape involved in the geometry shapes. And 2 means the size. So 2 shapes appear on the screen at the same time versus 3. And also D means the true answer is different. So the 2 sides in the study display and recall display are different shapes. And the same means they are the same. So even just looking at these box plots, we already see some different patterns that differ between the different groups. For example, we find generally the binding tasks are harder for the participants among all groups, but the MCI group considerably performs worse comparing to the cognitive normal group. And although size contributes to the difficulty. Another interesting finding we got is when the true answer is the same, generally the accuracy is higher, and we were I think one possible reason is that the participants, especially towards the end of the trials, gets fatigued and tends to select same other than different. So there is definitely a trade-off between the sensitivity and specificity here. So the main results I want to show today is from the multi-level functional principal component. And idea is to decompose the variability in the baseline adjusted pupil size as a function of t into different components. So the first component is the mean, the overall mean of the pupil size trajectory. And then the eta jt indicates the trial-specific shift from the mean. It can be set to 0 depending on the scenario. And then there are 2 types of variability in terms of the pupil size trajectory. The first one is a subject-specific deviation from the mean function, which is denoted by ZIT. And the second one is a residual subject and visit-specific deviation that is not explained by all the other components, denoted by WIJFT. So the MFPCA basically decomposes the last two items into principal component decompositions. And the first one is the level 1 principal component, which is the subject level principal component with the corresponding PC score. And the second one is the trial level principal component with the corresponding PC score. So this is the mean function of the binding task, and focusing on the 3 stages of study display, the blank period of 1 second, and then the unlimited time of recall display. And as you can see, the mean function of the baseline adjusted pupil size has a pattern that is low. The pupil size is small, considerably comparing the study display to the recall display. And then during the recall display, or even before that, it starts to increase and peaks at about 2 seconds or 1.5 seconds after the recall display starts. This is the first level FPCs, indicates the main modes of variability after subtracting the mean curve. So the first principal component explains about 75% of the total variability, which is given by a mode that is peaks at 2 times. The first time is almost at the end of the study display, so almost like overlapping with the end of the study display. So it increases from the beginning all the way to the end of study display and drops a little bit during the blank, and then increases again to the first second of the recall display. This is a major pattern in the subject level. And then the second principal component, which explains about 10% of the variability, is manifested as mostly a peaking pattern in the study display, and then drops down until the recall display and slightly increases at the end of the recall. But the major change actually happens about 1 second after study display starts, and also the beginning of the recall display. That's where the variability changes the most. And this is the second level, so the trial level principal component. The PC1 explains about 63% of the variability, which is mostly a change at the study display and increases and all the way remains almost flat between the end of study display and recall display. So we conducted a regression analysis just looking at the task accuracy of each trial, so it's either correct or non-correct, regressing on the different parameters of the task, including the set size, either 2 or 3, whether the answer is correct, is same or different, the true answer is the same or different, sex, age, and also the grouping using the normal p-tau negative as the baseline, comparing with all the other 3 groups, and also the level 1 PC1, level 1 PC2 PC scores, and level 2 PC1 and level 2 PC2 scores. So we found that the set size and the answer definitely contributes to the accuracy of the answer. Sex is found not to be significant in here in terms of the accuracy. Age is significantly related to the accuracy. And also the grouping, comparing the MCI, so both MCI groups considerably performs worse than the normal p-tau negative group. But between the normal p-tau negative and p-tau positive, there's no significant difference in accuracy. And finally, we found the PCs, the PC2 in level 1, and also the 2 PCs in level 2 are also associated with accuracy. So in the future, because of how rich the data is, we are developing multiple directions, including comprehensive stratified analysis, looking at the eye gaze and pupil patterns. And also we are aiming to integrate the two modes, the eye gaze, Y coordinates, and pupil data, and try to characterize the modes and rates of cognitive decline in different groups. And there is also an ongoing direction using deep learning methods to understand the patterns in both the eye gaze and pupil size, and for this kind of complex functional data. So finally, I want to thank all my collaborators and students at UCSD, and please feel free to ask me any questions. And also contact me at my email if there's any question you want to discuss. Thank you very much.
My name is Shilpa Limati. I'm from the NAC Tech team. Thank you to all the speakers for a very engaging, interesting first half of this data core workshop. Certainly from the NAC Tech team perspective, I enjoy listening to all this research and how the NAC data is leveraged. But I'm here for a happy announcement. We will now be taking a short networking and refreshments break. We will start again at 2:50 p.m. Eastern Time.
Today my topic is mean life and later life social risk scores, SRS, modify the effects of ApoE exon 4 on Alzheimer's disease. So, okay. So this study is a part of the project Comprehensive Gene and Environment Interactions in AD, which has been funded by NAC New Investigator Award this year. So I express my appreciation to NAC for this opportunity to share the project here. This project just started in August. 2 months ago. So here in the presentation, I'm gonna to talk about some ideas and aims and the very preliminary results that maybe inspire you. As we know, later onset Alzheimer's disease indicates people develop AD who age 65 years or older. So 60 to 80% load is heritable, according to twin studies. Multiple genes play a role in developing load. So ApoE is the very important gene, as we know. In addition to ApoE locus, a recent Jew was able to identify 63 AD-related single nucleotide polymorphism synapse in multiple genes. On the other hand, up to 1/3 load modifiable by environment factor. Many environment factors potentially affect load. So this list is kind of give you an example. So also environment effects on load may vary in mean life and later life. In the Livingston paper published in the Lancet 2020, they provide different environment risks during lifetime. For example, like hypertension is a risk factor in mean life, but could be a protective factor in later life. So many environment factor and multiple genes. Our question is how to comprehensively examine gene and environment interactions in law from a life course perspective. So current pattern were examined one-by-one interaction, one gene or one exposure model, or genome-wide loci and one exposure model. So moving forward one-by-one interaction, we propose to construct comprehensive scores for genetic risks and also for the environment risks, G and E. And that could be one possible solution to provide new insights. For many environment factors, we need a dimensionality reduction. There are other dimensionality reduction methods. We propose to use IRT, item response theory-based model. So what is IRT? I'm going to talk about a little bit about IRT. IRT is used to test scoring. So for example, the math test. So it is an assumption that math ability is indirectly observed, but math test scores are directly observed. So here, there are 5 items. Student 1 answered correctly 3 items, and student 2 answered correctly 3 items. For student 1, answered correctly 1, 3, 4. Student 2 answered correctly the item 1, 4, and 5. So they got the same total score, but we expect the student 2 may have a higher math ability than student 1 because student 2 answered correctly the most difficult item 5. So similarly, so we use environment variables which are directly observed to estimate environment risk as a latent variable, which is indirectly observed. So as well, we have 5 environment variables. So participant 1 answered yes to 3 of them, and participant 2 also answered 3 of them, yes to 3 of them. So they could have the same score, like 3 points, but we expect they may have different environmental risk scores because the different items could be different environmental variables could have a differential contribution to the latent variable. So IRT could help us to more precise to estimate the latent variable. So IRT model can be extended from binary to ordered response that allow us to handle different response pattern of the environment variables, and also from unidimensional to multidimensional that fits our theoretical framework. We assume the environment risk structure could be multidimensional, including like social, physical, lifestyle, or behavior factors. So, and we can estimate 1 parameter to 2 or more parameters. So that provide the property of the environment variables, such as the contribution to differential contribution to the latent variable. In this project, we apply multidimensional generalized partial credit model, GPCM, which were implemented by the MERD-R package. So back to the aim of this project. So we're going to construct a comprehensive score to represent the overarching risk using the polygenic risk score method. PRS is an estimate of a genetic reliability to low by aggregating genetic effects of a single nucleotide variant. And we also construct environmental risk scores, ERS. We instruct a mean life and later life ERS. And then we examine PRS and mean life and later life ERS in cognition and neuropathology. As a preliminary analysis, we apply GPCM to social variables that were from NOC UDS, the 3rd version data. So there are 6 social variables: preliminary language, education, marital status, and living situation, level of independence, type of residence. Because NOC UDS is a longitudinal, data is longitudinal. So we were able to construct, to use the participants' records at the age range between 40 to 65 to construct the mean life SRS. And we use participant record at the age range 65 years or older to construct later life SRS. And then we examined ApoE exon 4, the interaction between ApoE exon 4 and mean life and later life SRS in load. So 6 social variables were recorded for GPCM. So higher the SRS scores represent higher risk. So there's a percentages of social variable by race and lifetime. So we can see in later life, so approximately 40% of African American participants had a education year less than 12 years. And also 62% were unmarried, and 61% living alone. And compared to mean life, so the later life were about 10% more likely to live alone across race. So we applied GPCM to construct the SRS, and then we plot the contribution based on the factor loadings for each variable. So we can see among 6 social variables, 4 type of residence, level of independence, living situation, and marital status had a relatively larger contribution to SRS in the non-Hispanic Y and also AA groups. We visualized the interaction between ApoE exon 4 and the SRS on the Monchon cognitive assessment MoCA scores. And we find in a non-Hispanic Y mean life group, ApoE exon 4 carriers had a lower MoCA score on average. So in lower mean life SRS scores, ApoE exon 4 carriers had a lower mean MoCA score compared to ApoE exon 4 non-carriers. But there's no differences between ApoE exon 4 non-carriers and carriers in the high SRS group. In the AA African American later life group, so ApoE exon 4 carriers had a lower MoCA scores on average. We find ApoE exon 4 carriers who had a higher later life SRS had the lowest mean MoCA scores. So, which means in the African American participants, so who were ApoE exon 4 carriers with the higher SRS scores. So later life SRS scores may be more vulnerable. After visualization, we run the regression modeling analysis. We call ApoE exon 4 genotype as a additive mode. 0 represent the non-carrying exon 4. 1 means carrying 1 exon 4, and 2 meaning carrying a pair of exon 4. And we choose the phenotypes as outcome, the mini mental state examination, MMSE and MoCA, also A beta and tau, which were from the NOC neuropathology data. as a phenotype outcome. Multiple linear regression were applied to the neurocognition test scores, and ordinal logistic regression models were applied to neuropathology variables with the adjustment for age and sex. We conducted the analysis within two racial groups, non-Hispanic, white, and African American. So we got the regression results. In the mean life, so we can see in the African American group, there is a significant interaction between ApoE and also SRS. And in the later life, there are significant interactions between ApoE and SRS on MMSE and MoCA scores among the African American participants. What we found in the very preliminary results, I summarized, we construct mean life and later life SRS based on 6 social variables using the IRT-based model. And marital status, living situation, type of residence mainly contribute the mean life and later life SRS according non-Hispanic, white, and African American participants, which might indicate the factor so-called living condition. And we also found mean life and later life SRS modify the effects of ApoE exon 4 on the neurocognition scores, MMSE and MoCA scores in African American participants. So we need to verify the results in the future. And also, for the next step, we would like to include more environment variables, physical variables, lifestyle variable, and behavior variables. They're a variable from NOC UDS third version data. And we also look forward to UDS version 3rd data available maybe next year or 2026, so we could include more environmental variables, particularly social determinant of health variable. In the end, I want to give a big thanks to my mentors, Dr. Kasmada, she's here, and Dr. Fado, Dr. Nelson, Dr. Abner, and Dr. James, and UK ADRC Director, Dr. Linda Wen Aldick. Without their outstanding mentorship and support, I couldn't make it. And also, I want to thank you, our lab and members and families. I also want to thank, I have other funding resources to support my research that related to this project. So, thank you for your attention.
Thank you, Dr. Wu. We have time for one question.
Thank you very much for the interesting talk. So I really like the idea of using IRT to model the social determinant variables. Now, when you categorize the social determinant variable, did you make sure that they go to the same direction in the sense that higher score means worse or means better? So, or for all 6 variables? Because one variable is like a living, like a residence, or another one is the marital status, right? I don't know how you code it. I don't know, married is better or than the unmarried or vice versa. So did you make sure about that?
Thank you so much for your question. So I think this is a very important question, and I did check the directions for each of the variable. So.
Married is better than unmarried or vice versa?
Yeah, surprisingly, yeah, I'm married, had a higher MMSC and MOCA scores. Yeah, than married group. Yeah, yeah. So, so yeah, that's surprisingly. So probably, I think maybe living alone or unmarried, maybe will encourage people to can do more by their own. And probably they always kind of train their minds to learn new stuff and then being independent and try to learn more stuff to solve problems like themselves. That's one possible reason I can think about that. And I don't know other reasons, but the data at this point show me this pattern. I did check the directions.
So our next speaker is Dr. John Kornack from UCSF. It will be online. John is a professor of biostatistics at UCSF and the co-director of the UCSF Data Core. And he will talk about Bayesian multimodality disease progression model of dementia.
I'd like to talk to you today about our work on Bayesian multimodality disease progression modeling of dementia. And in particular, I'm focused on frontal temporal dementia here. Although I'm presenting this work, I am not the person that's done most of the work. That has to go to Melanie Quintana and Barbara Wendelberger at Berry. And also Adam Staffaroni and Adam Boxer at UCSF. So I'm assuming many people are not completely familiar with frontotemporal dementia, so I'm going to give an introduction to that. And partly I'm doing that because there are sort of unique problems that get presented in disease progression modeling when we're thinking about frontotemporal dementia. I'm always a little nervous talking about this in front of clinicians, but I'm going to take my chances. So FTD is an early onset fatal neurodegenerative disease. And it's estimated that about 10% of all dementia cases are frontotemporal dementia. So it's rare compared to Alzheimer's disease, but it's it's a particularly horrible disease. It affects people younger typically than Alzheimer's disease, as early as in the 40s or 50s. Now, one aspect of FTD that makes it more complicated for modeling is its heterogeneity. There are various kinds of syndromes that are related to the brain location where the disease starts. So there's behavioral variant FTD that leads to repetitive behaviors and inappropriate behaviors, language variants that can have different types, whether it's you're unable to find the words or you know the words, but you don't know what they really mean and things like that. And then motor presentations like ALS and PSP, CBS. So in addition to there being these different presentations, there's also another split in terms of familial versus sporadic frontotemporal dementia. So around 30% of cases are familial. These are autosomal dominant and of high penetrant. So if the gene is in the family, then there's very high risk of developing the disease. There are three main genes that we have in this. There are a few others, but they're rare. But the three main ones are progranulin, GRN, MAPT, and C9. And to add complication, these presentation of the proportions of the sort of, whether it's behavioral or aphasia or motor, varies by the genetic variants, but it's not as if one genetic variant has all of one type of frontotemporal dementia. Now, the remaining 70% are sporadic. That means that they occur, as far as we know, at random in the population. But the 30% that are familial are particularly important for research, because if we know that the genetic variant is in the family, then they can be useful for early stage clinical trials, and of course, just for longitudinal study. Now, we would like to use them in early stage clinical trials, but a difficulty that we have in FTD is that the heterogeneity of age of onset is very high. The data that I'm working with here is part of All FTD, which I'm a part of, and it's a longitudinal study that's now going in for renewal, looking at both sporadic and familial FTD longitudinally across multiple centers that you can see in the map at the top, and we collect all kinds of data, imaging data, cognitive tests, blood work, etc. And our patient age distribution is on the order of 40 to 70. The map on the brain maps on the right are there just to illustrate that heterogeneity that I was talking about. This is with respect to brain tissue loss, and you see so AD is in red, and then blue behavioral variant FTD, green semantic dementia, these different types of FTD, and you see that they're located at different locations around the brain where the tissue loss sort of originates. So we have challenges to conducting familial frontotemporal dementia trials. There's new drugs coming out, but how to design a good study is still an open, difficult question. It's based on what we've seen in Alzheimer's through Diane, and it's going to most likely be best to treat frontal dementia early at the pre-symptomatic stages. But in order to do that, we need good biomarkers of clinical changes, and we need to be able to figure out pretty well when their onset is likely to occur. But the problem is age of disease onset is highly variable, and that's within mutation and within family. See over on the right, it kind of shows you that roughly the distributions of age of onset for the 3 different familial FTD types. And we have as far as we know, no good predictors of age of onset. Disease course is highly variable across individuals. These are all challenges, of course, make our life difficult. Each symptom's onset is difficult to predict. So how do you set up your enrollment criteria for a study? Do we even want to have the same endpoints for if we're doing trials for different disease stages? And this is where hopefully the disease progression models can help us. So the first time I came across disease progression modeling, and I think probably most people in our field, was through the Cliff-Jack model for Alzheimer's disease, which I believe isn't really a model so much as just a kind of, it's a conceptual model. These lines are drawn to represent when the onset of variant aspects of Alzheimer's disease occur across the disease time course. So really the x-axis kind of represents disease age, and you see that amyloid beta goes up first, then you have the tau, brain structure, memory, and then finally clinical function. So we want to kind of have a good disease progression model that works well for frontal temporal dementia. And we want it particularly for clinical trial design. So if we can, by having these disease progression models, there's a lot of advantages. If we can have a good model, it's going to help us a lot with trial design. We can have targeted enrollment, making sure if we want to try and capture people earlier in the disease, we can perhaps predict them with a good model of what their disease age is. We can do model-based clinical trial simulations, so we can really start to understand what sample sizes we need for these studies. And it provides us with powerful analysis tools to understand when a drug might actually be slowing disease progression, if it's effectively slowing down the progression of disease age, in a sense. Just to make that a little bit more kind of concrete as to why these models would be useful here. If we were using predicted disease age for an optimal clinical trial enrollment, if we're capturing people too early in disease age, so like 10 to 5 years prior to their onset, then for the length of the duration of the study, their symptoms are unlikely to progress. And so we're really going to get no power in the study. On the other hand, if we're coming in too late and we're capturing people later in their disease stage, then by that point, the treatment is not expected to be as effective because they're well into the disease. So really, we want this Goldilocks sweet spot in the middle. We want to be able to predict who is going to shortly be progressing, so then we can enroll them at that time and capture the people that are likely to benefit. Okay, so finally, onto the models a little bit here. We're going to jointly model clinical endpoints and biomarkers over time. So we have a CDR sum of boxes score that's modified for frontotemporal dementia, various neuropsych scores, neurofilament light, which is a blood biomarker associated with FTD, and then volumetric regions of interest from MRI. And our predictor is going to be this disease age, which is really their chronological age minus their age of onset of disease, whatever we exactly mean by age of onset. And I'll tell you what our working definition is here in a moment. And we also have genetic group as a predictor, and just giving you a rough table of the numbers that we have. We have about 486, currently 486C9, 322 GRN, and 210 MAPT. And they're each have around about 2.5 visits per person. Okay. So this is our initial model for disease progression. Kind of walk you through it here. And on the bottom left, that plot is kind of what this model looks like. It looks like that S curve that was in the earlier diagram that I showed for Alzheimer's disease. And we have subject I, visit J, biomarker K, and mutation M. And the model structured as follows. So it basically has this S curve that's sort of driven by this part here. There's an endpoint value for each outcome. For each biomarker, we have an endpoint value at normal. This is determined from sitting down with clinicians. There's a subject-specific random effect for a change in whatever that normal value is. There's a Worst value for the endpoint, again, determined with the clinicians. There's a decay location parameter here. So it's kind of like your regression within the exponential term. The intercept here is an anchor. We anchor it to such that we say that disease onset, that means that you've got a disease age of 0, happens when your CDR score first, CDR sum of boxes first hits the value of 0.5. And then there's a decay rate. So how fast are your is your rate of decay in the disease, and disease age is defined, like I said, as age at visit minus age at onset. Okay. So this was our initial model. The hard part of all this is, you may have gathered, is the age at onset part. And if the onset, even if the onset is observed, clinicians don't feel like they can give really reliable estimates. So we model that as a priori normal, with mean being the clinician estimated value and a standard deviation of 4 years. Again, this was sitting down with UCSF faculty in neurology and figuring out what an appropriate prior is. And if the onset has not yet been observed, it's modeled as normal with the mean for the mutation type and a standard deviation of 10 years. So a much larger standard deviation in that case. And finally, for non-carrier family controls, we have that the disease age is based on the mean age of their family's mutation, again, with a wide standard deviation. Okay. So just looking at time, and I think I should be wrapping up pretty quickly. So I just want to, these kind of like illustrates the kind of results we get. So with top left, we have the CDR sum of boxes score. Here's an individual test that trails B. Bottom left is a volumetric frontal lobe volume. And bottom right, the NFL biomarker that I mentioned. And you can see for the CDR sum of boxes that the progranulin group seems to accelerate much faster than either C9 or MAPT. These are anchored, as I said, because to that 0.5 being the onset time. But you can see with the other, so for example, the Trails B score, that the onset for C9 occurs, or the sort of like the beginning of performing more poorly with Trails B occurs before the official disease onset time. Okay, I'm gonna just mention very quickly about our model that we're now pursuing. We're extending the model a little bit from what we had originally, partly because we didn't like the symmetry that was induced by the model that we had previously. Basically, the rate of progression was the rate at which it approaches its maximum, and that seemed like too strong a constraint. So we've added in a change point here. Here is pretty extreme, but as an example, so you can have a slower rate of the rise and then a much faster rate to the top, and that sort of comes from this middle part here in the model extended. We also bring in a subject-specific random effect for what the individual rate of increase is. Okay, I'm not going to go through the pictures. I'm just going to kind of come up with some final comments here and saying, like, you know, these models are kind of, it'd be great if we can get good fits to these models, but there is a little question of like, maybe we're asking too much of the data based on annual visits. And that's something we're trying to get around with our future studies. Are we really going to have enough information to pick apart within versus between subject trajectories? We need to do, what else can we do to validate our predictions? It's one thing to come up with predictions of disease age. It's another to actually know that what we're doing is reliable enough to come up with a power calculation. And how do we improve our determination and or definition of what onset time is? I mean, that working definition of hitting CDR sum of boxes of 0.5 is kind of something we can use, but it's not really, is it really meaningful? And with that, I'll stop. Thank you very much for your attention, and sorry again for my technical problems previously. No problem.
The next section will be the updates from the electronic data capture work group, and we're all eager to hear. And we'll start with Dr. Sarah Bieber. She is the executive director of NAC, and she has been leading the effort of modernization and expansion of NAC since November 2021. Welcome.
It's actually Bieber, but Justin Bieber ruined my last name, but hello. It happens all the time. It's okay. It's fun to, I'm not related, by the way. No. So I want to thank Iris and others on the NAC's Data Core Steering Committee for planning this great Data Core workshop today. Thank you so much. It's been wonderful to be here with all of you. Yeah. And I'm honored to join you here for the tail end to share some of the outputs of our amazing, really amazing collaboration with the Electronic Data Capture Workgroup. So I'll be joined by Jess Welch from NAC and my colleague Chad Murchison from one of our newly funded P30s at University of Alabama. So over the past couple of years, we have been working together to make it easier for ADRCs to collect, submit, manage, and access participant data. I want to remind everyone, still can't do it, won't let me advance. I want to remind everyone that building a modern electronic data capture system for the ADRC program was a big socio-technical challenge. When we first got started, the existing system was 20 years old and needed to be updated to be more cloud-friendly and virtualized. ADRCs were doing data collection 37 different ways, and we had some big goals for our new electronic data capture system. So we wanted it to be technically modern. We wanted to develop powered by REDCap to effectively serve the needs of the ADRC program. We want it to be robust and scalable to numerous data streams in addition to UDS. And we really wanted to use this opportunity to really foster collaboration across the ADRC program software development community and data cores. And we also wanted to promote and share best practices and make data-driven decisions as we went along. So our EDC work group was launched in January of 2022. We've had 111 people from across 33 ADRCs involved in this group since we got started, many of whom are still actively involved today. You can go to nacdata.org/EDC to learn more. And our new system is really designed to save time, enhance sharing, and improve data quality through this new electronic data capture and submission system. So it provides ADRCs with a range of data collection and submission options. So for collection, ADRCs can use their local REDCap. They can use an alternative local EDC. They can continue to use PDFs. They can do direct data entry into NAC's REDCap instance. And for submission, they can upload data via an API or with bulk CSV, or they can do automated submission. So we've designed a very flexible system. It has a host of different options. And I'm excited, we've collaborated with the EDC on all of these ones that are starred here. So they've been absolutely critical this collaboration is absolutely critical to rolling out all of these critical new systems for the ADRC program that are not only going to set us up for success with UDSV4 data collection submission, but also with many collection submission of many other data streams down the line as we become more multimodal. So I want to give a quick recap of some of our other major accomplishments as a group. So our development sub-team we've now completed 100% of the UDSV4 packet. We've developed REDCap forms for all of those. Amazing work, everyone. We've established the infrastructure for longitudinal data collection. We've established a GitHub repository with UDSV forms and resources. And we've collaborated on a new NAC ID process, which my colleague Jess will share more about with you, for flexible multimodal data collection. Our requirements group has harmonized UDS data flow for ADRCs. We've built a programmatic and human-readable UDSV3 to UDSV4 data variable crosswalk, which you'll hear about from Chad. He's been instrumental in that. And we've streamlined and standardized the REDCap submission process, quality checks, and variable names. We've also deployed data management plan in alignment with NIH's new data management and sharing policy, which is available on our website, and you can use as a starting point for developing your own data management and sharing plan as you submit your own grants at your ADRC. Our documentation and training group has launched a NAC EDC documentation website, which you are encouraged to visit, with comprehensive technical documentation resources for UDSV4. They've also developed SOPs to facilitate UDSV4 collection and submission, and established an ADRC program community forum powered by Discourse. My colleague Elise was really instrumental in getting that going. We've also collaborated on the UDSV4 resource landing page on the NAC website, which you're all going to see a lot of links to over the course of the meeting this week. So essentially, we set out to streamline and reduce burden with EDC. We've been very successful at that. Some of the ways that it saves time is allows direct data entry. We have autofill drug codes that are available through the RxNorm ontology. You can auto-populate health history data for follow-up visits. There's skip logic built in. You can have access to automated NAC tools that are connected to data. And you can also have access to on-demand centralized training. In terms of enhancing sharing, we've made some big headway there as well. We've improved a solution for co-development and sharing amongst ADRCs. The forum is one important piece of that, our GitHub repo and other resources as well. We've conserved resources through centralization, so we've enabled other ADRCs to learn from each other, right, and to leverage each other's resources. And then we've developed automatic EHR and REDCap extractions for ADRCs to help facilitate the data collection submission process. This work has also helped to improve data quality. So the real-time error checks help with that. We've reduced multiple choice errors. We have transparent and replicable QC processes. They're all available on our REDCap repo. We've improved standardization and error checks through centralization, and we've also enhanced utilization for analysis and other scholarly activities. So we're really, really proud of the work that we've accomplished together as a group, and I think this is a good moment to step back and just reflect on that. So a couple other things. We have been successful at driving REDCap adoption across the ADRC program. So you can see here over the course of our work together and the course of our many surveys, thank you guys for responding to those so well, we have more and more adoption of REDCap happening across the ADRC program. And we will eventually, down the line, require that all sites use REDCap. So we're getting there. It's not going to happen for UDSV4, but that is definitely where we are headed. So you're going to hear a lot about this at the next session. This is like my favorite figure of the whole meeting. But UDSV4 is launching. It's happening. We have a plan. We have consensus around a plan. Many of you helped inform this plan with the feedback that you provided on our UDSV4 pilot. And UDSV4 is going to be available to launch at your site starting in January of 2025. In addition, you will have 6 months to launch UDSV4. You'll be required to have launched it by the end of June. So we'll talk more about that. You get to hear all about it on Thursday, but I want to give you guys a preview of coming attractions. And one of the things that we'll be doing as part of this is actually launching another real-time dashboard at NAC. And so we'll be tracking in real time. This will be a public leaderboard dashboard to track submission of UDSV4 data across the ADRC program, both initial packets and finalized packets. And we're doing that for UDSV3 as well because we need to, of course, wrap up collection of all remaining UDSV3 packets over the coming year. So that's all coming. You'll hear more about it, but you got a preview here today at the workshop. And we have developed an incredible range of resources. Enormous amount of thought and care has gone into developing these, not just by NAC, but by our electronic data capture workgroup and by this clinical task force to provide for all of you. So these are available on our website. Once again, you'll see this QR code a lot this week because we really want to drive traffic to this site. If you are feeling overwhelmed by getting started with UDS v4, I really feel quite confident that you're going to find resources here that are going to allow you to get started, and we're here to help, so please reach out with any questions. Lastly, our work is not done. So if you want to join our EDC workgroup, it's not too late. We'll have a sign-up information on that in a minute. We still have a lot to do, so we have some follow-up visit packets that we need to develop. We have additional modules that we're going to be developing in REDCap for FTLD, for LBD, for Down syndrome, and other data collection forms, like our neuropathology form, BDS, and CLS. We have new REDCap form for digital voice data collection that we're going to be developing, as well as for integrating, developing some resources for integrating data from multiple data streams to assign ATN biomarkers to UDS v4 participants. You'll hear more about that from Gil Rabinovich later in the meeting. And we also have REDCap forms that are going to be developed for Spanish and Chinese translations of UDS v4. So lots more work to do. We're excited to bring in more partners. This group is open to everyone across the ADRC program. So you can join, if you're interested in learning more about joining the EDC, you can go to our website. You can use this link right here to join. Like I said, it's not too late. And lastly, I really want to thank especially my co-lead, Sudeshna Das, who has been an amazing partner in getting this group launched and keeping it going. I also want to thank Jess Welsh, who has, since she joined NAC, really helped to amplify the efforts of this group even further and keeping us all organized and on track with all of these amazing deliverables. I also want to thank all of our other leads, especially, and all the members of the group, of course, but Meredith Zozis and Chad Murchison, Carrie Stevens from NAC, as well as John Reeder, Debbie Zemlock, Catherine Gothrow, as well as some of our Laura McLeod, and also one of our newest member, Naranjana Shashi Kumar. So thank you guys so much. This has been an amazing collaboration, and we're really looking forward to next steps with all of you.
My turn with the with the remote. Hi everyone, my name is Jess Welsh. I'm the Senior Project Manager here at NAC, and I have had the distinct pleasure of working with our very hardworking members of the EDC over the last my last year here at NAC. And I'm going to talk to you a little bit about one of our central tenets here at NAC as we continue to build out solutions and deliver new exciting advancements. So one of the things that you hear over and over from us is that we don't want to build something and hope that people come, right? We want to build iteratively and involve our stakeholders at every single part of our journey so that we're creating solutions that are relevant, that will stand the test of time and be scalable. So one of the biggest things that we've done over the last year to demonstrate this is our pilots. So we conducted 2 pilots over the last year. Many of you were involved and got to participate and provided feedback. There was the UDS v4 pilot as well as the NAC ID pilot. I'm going to give you our key takeaways from those today, and then we're going to talk about the NAC ID rollout. So we're going to start with the UDS v4 pilot. There we go. All right, so over the summer, some people spent their summer working on a UDS v4 pilot, which was launched in order to be able to test the new UDS v4 forms, as well as the updated ADRC portal and submission methods. So it was a big pilot. 23 ADRCs participated. 16 of those tested those forms on clinical core participants. 1 did mock participants. Overall, 55 subjects were administered the new forms, and they spanned all of the cognitive diagnoses. So you can see here from this slide that the pilot sites tested any of the collection methods, right? So all the collection methods that Dr. Biber showed, the direct entry into the NAC-hosted REDCap, which is a newly available feature for submitting data. There's also direct data entry into their own local REDCap instance or their own local EDC, and PDF forms were still also very popular. Sites were also able to test all of the new submission methods, or all of the submission methods that we're planning on launching for UDSV4. So direct entry, again, into that NAC-hosted REDCap, batch upload via CSV, which is the most similar to how sites are currently uploading for UDSV3, and then that automated, very popular new feature, API submission through programmatic upload. So what we found, thanks to all of your feedback, was that sites used many different resources in order to be able to navigate the forms and to navigate the submission methods. One of the most popular was our ADRC program community forum. So many people tested out the ability to go in and connect with both NAC as well as each other on our public forum. Also popular was the, of course, data element dictionary and the submission demos that the CTF has created. Those were all available, distributed via email for the pilot, and will be online going forward. So, if you're wondering, right, this is new forms, new submission methods. How easy was it? How hard was it? The good news is nobody said it was very difficult. So, most people said it was somewhat easy or very easy or neutral at this point. There we go. All right, same thing with the new submission options, right? So an updated, totally new UDSV4, ADRC portal. And most people said it was, or most of our respondents said that it was somewhat easy or very easy to be able to submit their data using the new portal. And one of the other findings that we found that was really nice was that even novice users of REDCap, so for those of you sites who are just starting to use REDCap now, still 75% of you found it was somewhat easy to be able to submit data using REDCap. So along with all of these quantitative numbers, we also got a lot of qualitative, a lot of tips, a lot of ideas, a lot of feedback from you, and we wanted to just highlight a few of the key changes that we're making in response to your feedback. So one of them was slight changes to the REDCap form logic and rules that was set up by the EDC's development sub-team, and so those are being modified slightly in response to your feedback. Another one was making the API, or the programmatic upload documentation, a little more accessible. So people asked for more examples of a demo video, so all of those are going to be available on our website going forward. And the other thing was having a sandbox to be able to test in. So we did have that available for the pilot, but we are extending that and making that available to all ADRCs for the foreseeable future. All right, so I'm going to end the UDSV4 pilot talk there, and we're going to switch over to the NAC ID pilot. So a year ago today, we were in a different room, and I announced NAC's plans to launch a new NAC ID process to be able to assign NAC IDs at the time of consent. This is critical to being able to use or to upload multimodal data and be able to have these connections across different data streams. So going forward, once the new NAC ID process starts, once a participant is consented, they will fill out the NAC ID form via REDCap or CSV upload, and then those NAC IDs will be available immediately. This is super important to people like ADRCs, but also our affiliated studies and partner organizations. So this pilot had 32 ADRCs participating, and again, tons of feedback, 44 feedback surveys were returned, and this group really jumped on the community forum dialogues, which was great as well. So this is an entirely new process. It's a change in workflow, it's a new form from NAC, and still, 83% of our respondents said that the submission methods were, the submission methods were easy or somewhat easy, so that was good. The other thing that we saw from you all when you sent back your feedback was that 97% of you said that you felt prepared. So the resources that were provided and the instructions that were given felt prepared, or mostly prepared, and that included the API as well. So the changes that we're making to the NAC ID process based on the feedback that we'd like to highlight are, one of the things we really heard was making help documentation more visible and accessible was important. So we've inserted links throughout the portal, as well as consolidating our resources online as opposed to emailing them out. So you'll see that in the adjusted process. We've also displayed or added a new display to the process that shows your uploads, and you can see your NAC IDs. That wasn't available during the pilot. You're going to get to see it today. And then we're adding more submission documentation, including videos for the API. All right, in the great words of Sarah Gothard, I'm going to allow myself to introduce myself for a demo video. Maybe.
In this video, we're going to walk through the Managed Participants segment of the ADRC portal. In the Managed Participants section, we are able to assign new NAC IDs and transfer existing participants. When we click on the button, we are taken to a page where we can view our existing participant identifiers. We can view enrollment uploads that are in process. For this video, we're going to go through the submission process. So if we scroll down, we're able to choose to submit information to generate new NAC IDs through bulk file upload, direct data entry into the NAC hosted REDCap environment, or upload via API. So for bulk upload, we're going to start there and just click on that button and click to proceed. This will take us to an upload window where we can simply drag and drop a CSV with the necessary variables and correct suffix on the file. We'll click upload. And from there, we're taken to our page where we can see a summary of all of our recent uploads, how many versions there were, and what the QC status is. We're able to launch a quick pop-up showing what the errors were. And then we can also go to the full error report where we'll be able to see all of the detailed errors and alerts that were generated by our upload, and then we can download them for future use. Also on this page, we're able to go to some important quick links like back to the upload page, to REDCap direct entry, to the ADRC program community forum, or to our electronic document capture page with documentation. So that is the Managed Participants section of the ADRC portal.
Okay. So, for those of you who are very excited now and cannot wait to get started, let's take a look at when you're going to get your hands on this. All right, so we've gone through the first two phases, right? We announced it a year ago. We did development and testing over the spring and summer. Now we're in the preparation and training section. So we are going to have an informational webinar that's going to show all of the new features based on the community feedback on November 13th. Final documentation will be available in one spot on the website, there for you to be able to access. And then there will be a period of time, just like we're planning for UDS before, where you can get your staff ready, you can do your internal preparation and training, and then for New Year's, you get to start using it, and it will be required that when you consent a participant to this study, that you will also, at the same time, send in the enrollment form to generate a new NAC ID. And then that NAC ID will be available immediately for that participant going forward. So that's the word. All right, thank you everyone. Thank you so much to our EDC and everybody for helping.
Spend 18 hours a day working with technology, and this remote is scaring the hell out of me. Oh, it works. Okay, thank goodness. All right, so my name is Chad Murchison. I am with the University of Alabama at Birmingham Alzheimer's Disease Research Center. I want to begin by taking advantage of the fact that I get this podium for a moment by commending Rachel for presenting earlier today. She was willing to present her own research on a dais that was chock full of PhD and core directors, and she more than held her own. One of the things that we did when we submitted our P30 was actively write in a position for a graduate student within the data core. We often hear about how the clinical cores take central roles in training early career clinicians for Alzheimer's disease, and I see no reason why we can't do the same with data. So I think it's very important that we sit here and we prepare the next generation of statisticians and data science and ADRDs, and I would encourage you, as you're preparing your own renewals, to consider doing the same. So that being said, let's talk about Harmony. So, yes, all right. So what is this crosswalk project? You have probably heard murmurings of it in the discourse forum, or maybe talked about among leads, among the EDCs. But what is the goal of this crosswalk? It is essentially to create a comprehensive mapping between the variables in UDS3 to what we have been able to identify as their most proximal counterparts in UDS-V4. And in this goal, we actually have two variants of the crosswalk that we wanted to provide. The first of which is a human-readable variant, one that is designed for easy annotation, something that can be modified by individual ADRCs, and is really going to provide all the structural changes and content, as well as potential mapping roles that we've been able to identify. But that being said, we are not barbarians. We're not going to sit here and make these mappings by hand. We're going to let the computers do it for us. And on that front, we have also worked towards developing a series of JSON files that can be actually be leveraged computationally and programmatically that are going to be agnostic to computational settings. So multiple programming environments can all make use of these JSON files. In addition to simply providing these crosswalks from a structural standpoint, we also want to make sure that the centers have enough agency to make modifications and develop them for their own unique considerations. So for that, we want to be able to provide tools that are going to be able to take those human-readable tables that you have modified and essentially regenerate JSON files that are going to be specific to your individual ADRC. In addition, we talk about JSON files and crosswalks and R packages, and we know that can be very obtuse and a little intimidating for folks that are not intimately familiar with their layout and utilization. So we also want to make sure that we're providing a lot of support, documentation, and guidance on how these tools can best be utilized. Bueller. Come on. Okay, so why do we want to do this? So it's, again, to provide structural correspondence between everything that exists within UDS3 and everything that is coming to fruition with UDS4. So we want to identify those variables that have been added, those that have been removed, and we also want to track where there have been changes, how have responses altered between the two variants, and what kind of things do we have now in terms of like question labels, metadata, positions within the UDS form, conformity, what are the allowable values? These are all things that we want to be able to track and appropriately map. So the reason that we're doing this is to try to make life easier for all of us, as I sit here just stating wildly, as we make this transition to UDS4. This is a lot of work. It has taken years simply to get to this point. And Sarah's saying here, all right, guys, 6 months, you got this. And we want to try to make this as feasible as possible because we understand that it's not simply a matter of collecting this new data and uploading it to NAC. You make use of this. You actually utilize it for a variety of purposes. And we want to make sure that you're able to continue to do this as you're slowly integrating UDS4 into your current data collection paradigms. So we want to make sure ultimately that we're minimizing disruption for the individual centers. That's really what the goal of the crosswalk is. So on that front, we're essentially covering 3 main domains. The first of which is going to be syntax and format. So this is very much that verbatim phrasing, the very specific things like, oh, look, there's an HTML tag here. Maybe we should take allowance of that as we sit here and make a new label. We actually have the structural changes, which are kind of the most important aspect of the crosswalk here. So again, the conformity of allowable values and responses, as well as how the forms are being organized. We all know, for example, that the A5 and the D2 are getting combined into a single form. Meanwhile, the Z1 form no longer exists. And then finally, this is going to be the first pass at actually trying to align on semantic meaning. And this is really where we get to the distinction between a crosswalk and a harmonization, which I'll talk about a bit later. Okay. So again, there are multiple functions here, and this really is a novel resource that is unique to the UDS4. Those of you who bore witness to history going from UDS2 to UDS3 know that it took about a year for a lot of centers to make this through because things like this simply didn't exist, at least not in a very uniform setting. And we're hoping to really kind of change that here. So we've mapped out all these changes on a form by form and item by item level that we've been able to identify from UDS3 to UDS4. Again, this includes things that are being removed, things that are being added, as well as actually seeing how things changed and how much agreement there actually is on a form by form basis. So I talked about the form dependence out of this, and the thing is, it's actually very nuanced and complex, and it really is best to think about this on a form by form level. We know that a lot of the forms are really seeing no meaningful change. So this is going to be things like the GDS, the FAIS, the MPIQ. These really aren't getting adjusted in terms of the way that they're being collected, the way they're being recorded. We have a number of forms that are essentially seeing, well, very minimal change in the grand scheme of things. We have our 3 new forms, social determinants, the tracking of immunotherapies, as well as the reintroduction of the UPDRS. So those don't have origins within UDS3, but it's still something that we want to allow for. We have some forms that are seeing very minimal change. So the A2 for the co-participant is actually just seeing a severe winnowing in terms of what's actually being collected. It's a much smaller form than before. The A3 for individual family history now includes a secondary diagnosis for the family members. Genetics has been removed, but it's still super wide. So yes, we are getting 25 siblings and things like that still. Consolidations are important because we're trying to make remote visits more accessible than ever before. And so now the C2 neuropsych form is being consolidated to cover both the C2 and the C2T to really make it easy to transition from one to another and then back again, as well as the inclusion of the new verbal learning tasks, so the RAY and the CIRAD. And then I'll talk about the A4 a bit more. That's kind of what I'm considering unmappable because we're going from our legacy identifications of medications that go back to like 2012 to something that's much more modern, making use of the RX norm ontologies, which is just oh so beneficial. Then things get a little more complex. So we have things like the A1, which is now including race, that is being vastly expanded to our benefit. We have the D1 being split out into a clinical diagnosis as well as a biomarker diagnosis specific variants. Then the B8 and the B9 for the clinical and neurological examinations are getting a number of malleable medical definitions to coincide with the updates that the field of ADRD has seen over the last several years. As mentioned, the Z1X is kind of being distributed out, a little dispersed. And then we have something that I would say is probably the most notable change out of UDS4, which is the consolidation of the A5 and D2. Importantly, in addition to these kind of mergings of medical conditions and health history into a single form, we're also reaching a point where ADRC decisions need to be made. So it is going to be up to you to decide, for example, how is it that I actually want to consider mapping repeated blows to the head or traumatic brain injury, because that's vastly different between what we see in UDS3 and what's being done in UDS4. And you need to decide what's going to be important for you, because we know, for example, Boston is going to have much different considerations than what we have down in Birmingham. So I mentioned race, and this is actually one of the most notably complex examples because we've gone from having a single ethnicity question as it relates to Hispanic origins and the idea of primary, secondary, and tertiary race into kind of a many selections, as many as you want, with 7 different categories on the whole. Some of these are easy to work with. So we have ethnicity, we have ethnicity, which does a nice bidirectional mapping between yes and no of being Hispanic, as well as individual mappings for specific ethnic groups that actually do map very conveniently between 3 and 4. as well as the idea of other. So we do have some consolidation. So all the values within UDS3 do not actually map to UDS4 at this point. But that's just the easy low-hanging fruit of ethnicity. Now we also have considerations of what to do as we go from the idea of 3 different races that now need to map to a single location. For example, we can have Black or African American from primary, secondary, or tertiary, but now they only map to a single response within UDS4. Similarly for Asian, white, et cetera, et cetera. But then we also have situations, for example, we are now expanding this beyond the census definitions to also include things such as Middle Eastern or North African, which is mappable from others, but it does involve the use of, say, regular expressions or text matching. So it's not quite as clear-cut as you would expect to be in terms of a one-to-one correspondence. And then by the end of it, this is only a fraction of what we've seen, but we're already starting to look like the MBTA subway map at this point. So you can understand how incredibly complex this can actually become. So again, for what we're providing, there are 2 main things that we have. The first of which are going to be the human readable tables. So within those, we have 3 data dictionaries in total. We have the UDS3 data element dictionary or researcher's data dictionary consolidation listing the forms, responses, and so on. And then we have a similar variant that is exclusively using the provisions that were on the NAC website for the REDCap implementation of UDS3. In addition, we then have those same components within UDS4. So even if you don't make use of the rules, you do have consolidated data dictionaries that you can make use of. In addition to providing those, we also indicate where change has occurred that we've been able to identify at an item-by-item basis for everything that I've just talked about previously. In addition, we then go through and list every identifiable mapping that we've been able to find between UDS3 and UDS4. And when I mean everything, I mean everything. So number, position on the form, the verbatim text of the label itself, where responses match in terms of both their factorial level as well as the actual label that responds to them. In addition, we're giving notice, for example, I mentioned things like the A5D2 that may need some ADRC specific considerations. So if we have identified that we need a little more guidance, we can't give you everything, we indicated where those rules are. And then we also realized that there are some things that don't just give a one-to-one mapping. For example, that Middle Eastern North African racial group. And so when special rules are needed for structural, partial, or conditional remappings, we've included those rules as well. So this is what the dictionaries look like on the whole. This is a subset of, yep, this is the A1. So we can see some, yeah, it is the A1, sorry, it's tiny text. So you can see how we have a whole lot of components giving the individual, let's see, it does work. So sorry for the folks watching online, you can't see my little green laser, but we have all the data dictionary components over here on the right, as well as those crosswalk change indicators that we've been able to identify over here, as well as what we've identified as a special mapping within the context of REDCap specifically. In addition, we also have the item-by-item mapping rules, and so every little bit that we've been able to identify has a listing at this point, and these tables can be modified by individual ADRCs, and you'll learn more about how to do that in future webinars. And then these can be used to generate JSON files as needed as you move forward and try to actually apply the crosswalks to your own data sets. In addition to the human readable variants, which are going to be useful for everybody, we know that the JSON files are going to be what's most critical for the folks here in this room. Again, we're focused on JSON files because we want to be very agnostic to whatever computational settings you're actually using. If you use R, keep using R. If you're using Python, don't change on our account. The JSON files are very universal, and this is why we decided to focus on them. We give a mapping from a UDS3 variable or variables to the approximately matched UDS4 variable, including certain priorities that may be needed. Again, think the A5D2. We've then done the mapping, and it's organized by type as well as by nesting. Anyone who is familiar with a JSON file will understand what the nesting looks like. And then as needed, we do actually provide computational and programmatic codings. So a lot of if-else statements for conditional mappings based off of responses within a form or across forms, as well as certain equations. The best example that I can give of this is thinking about how we're going from some diagnoses that actually had year within health history and now are based off of age. You can do those mappings, you just need to do a little bit of math or more accurately have the computer do it for you. And this is a JSON file. If you've seen it, you've seen it. If you haven't, get used to it because we're going to have a lot of these. Okay, so what is the scope of these crosswalks? The important thing to mention for this is that these are strategic support that is designed for internal use by the ADRCs. Okay. So again, we are trying to minimize disruption for centers themselves, and I'll kind of talk about why this is distinct from what you may be getting from NAC in the future when you want to actually do research. We are emphasizing in this structure a red cap to red cap matching. Okay, that really is what we're focusing on the mapping. It's what the EDCs have been working with. It is what NAC is trying to encourage centers to use. And it's what we have access to in terms of the data dictionaries. It was just the easiest way to get this out into the wild. So again, the idea is we want these tables to be modifiable. And so if you need to generate a JSON file ad hoc, you can absolutely do so. And again, that curation of special rules for non-conventional mappings can be modified or adjusted or tweaked at your convenience. The important thing about this, though, is that these crosswalks are actually the first step in what's going to ultimately be a broader harmonization of all the UDS data, where we bring in everything from UDS 1 to UDS 3 and then combine it into UDS 4 so that we have good harmonized data sets moving forward. And it's at this point that we actually do need to draw the distinction between a crosswalk and a harmonization. So when you think about a crosswalk, you're really thinking about relating variables to each other. You're thinking about that structural alignment and that structural content. How do responses actually map? Do we need to do adjustments on codings for individual variables and things along that line? The emphasis here is on format and syntax. And again, with our current context, we're thinking about this for operational purposes within an ADRC. This is very different from a harmonization, which is now designed to make a data set compatible and comparable with another data set. So globally, think of something like combining ADNI and NAC data for the UDS. What does that actually look like? This is going to be very similar because now we're going from UDS 3 to UDS 4, and mapping all of these together and actually harmonizing them does require a gentle hand that is different from that variable level implementation of the crosswalk. So transformation of data may be needed. It's going to unify things that used to exist. For example, we had the UPDRS in UDS 2. And again, the important distinction here, I think, is that we are now aligning in terms of the semantic concept. What does the data mean, and what does it actually represent, and not just like, what is it that is being collected? So in this purview, it is now being used for research purposes across ADRCs, and not solely to improve operations within an ADRC. So this came first mainly because the field has changed and we have new clinical priorities and we need a clinical viewpoint for a harmonization. We can't do this solely based off of the data. But we had to start by identifying those broad structural changes, which is where the crosswalk comes into play. Again, the goal of the crosswalk is to make life easier for all of us. We're trying to minimize center disruption during this transition. So we want to make sure as you're doing clinical consensus conference for people that you've been following for 5 years, you're able to smoothly integrate their UDS4 visits into all of the previous data that you've collected at this point. Some of you go back decades at this stage, and we want to really kind of minimize that. So again, this data focus is preceding the semantic meaning, but this does serve as a very easy and intuitive first step. So ultimately, the crosswalk is going to be the foundation for the harmonization. It's going to be a deeper alignment of the data itself, and again, mapping concepts as much as content. The important thing from this as well is that we can also start to think about variable derivations. I talked about before how the A4 is kind of unmappable because we have RX norm ontologies versus the NAC ontologies. But if you've taken time to actually look at the researcher's data dictionary, you'll know that the A4 that you get talks about classes of medications. And this is something that we will be able to harmonize between UDS 3 and UDS 4. So again, we can formalize these clinical and medical concepts, and we get good variable correspondence as a lead-in for this comprehensive standardization. So again, task force is going to be aligning NAC as a coordinating group in terms of representing what the data is. We're going to have input from the ADRC data management and statistical cores for how the data is being used. And then again, importantly, bringing in the clinical cores in order to make sure that we understand what the data means in terms of those clinically oriented concepts. So things to look forward to. Right now, you can actually go to the website and you can find those human readable tables that I had mentioned. So they are a bit of a first draft. We are waiting for the finalization on the pilot material from UDS4 in order to do an update on them, but we wanted to get them out into the wild. We wanted to make them available to you to be able to start investigating them, because something that you can do to help is give us feedback. We've been staring at these a long time, and we've probably missed something. There could be ways that we could improve the layout or the format, and we really want to hear from you on the discourse forum or from email on things that could be improved. We're going to have a GitHub repository to allow for that JSON file development, so you're not beholden to whatever it is that we put up. And then we're going to also provide webinars on how to actually utilize these. And then finally, there is going to be a call to action where if you want to get involved with the EDCs, you can, but soon we're going to have a harmonization task force as well. So again, if you want to learn more, there's the GitHub or the QR code for all the material that we have for this, and expect to see more in the future. Thank you.
Okay, we are going to ask Chad to take a seat, and our additional speakers to join the stage, Jess Welsh, Dr. Biber, and Dr. Doss.
Okay, any questions?
Hi, I'm Bud Kukel. I just want to make a comment that this is an overwhelming amount of work that's been done in order to pull all this together, and it really moves NAC and the data to a whole new level that hasn't been reached before as we move from one version to another. So I really just want to applaud all the work that all of you have done. It's been it's really amazing. Thanks.
Thanks, Bud.
Thank you, Bud. You know, we started this concept of this collaborative working group between NAC and the ADRCs when I was in the Data Core Committee in 2022, right?
We launched it in 2022, yeah,
Yes,
January.
Yeah, so when we launched it and never had this idea that this would take wings and be where it is today, and much of that credit goes to everybody who is volunteering their time from the ADRCs, like my colleague Chad over here, and many of you, as well as like the incredible NAC team that Bud, you have hired over the years and put together, and a special shout out to Sarah. This wouldn't have been possible without like Sarah's leadership and the incredible NAC team.
I just wanted to pipe up with some virtual questions that came in throughout the presentations. So the first question came in during Dr. Biber's talk. This question is, when will the centralized REDCap for UDSV4 be available? Is it January 2025?
So many of you already tested this out via the pilot, which was wonderful. We are going to be continuing to host a sandbox environment where you can jump in and continue to test this out. All centers are invited to officially start collecting UDSV4 data for their participants starting in January of 2025. So all the REDCap resources will be available for you to do that. Then officially going into the database, if you'd like to practice, you can continue to practice in our sandbox environment that we set up for the pilot. There's more information that will be being made available on our website about that very soon if you follow the QR code that we've been sharing.
Great, thank you. My next question from an online participant is for Jess. And the question is, do we have all UDSV4 REDCap instruments in the GitHub?
The UDSV4 REDCap instruments will be available in 2 different ways on the centralized website that you're seeing the QR code for everywhere here. So the NAC UDSV4 updates page. There will be individual zip files for each instrument, and there will be an XML file that will have all of the instruments together.
Any other questions from the room? Yes.
Dan Hung from UCI. So I have a question about the BDS and non-UDS4 forms. So one of the pain points is we have to manually enter the BDS. So is there a timeline for the automatic API for the BDS and also like the Down syndrome modules? If there's anything, I'd like to volunteer to like push that forward.
Well, I would say if you'd like to sign up, we would love to have your help. We do not have a timeline broken down for the individual additional forms that we're going to be doing yet, but that is coming soon. And we've had some amazing folks involved in this work group all along, including some new folks that have joined us recently as a few weeks ago, and we're, have some folks who have been there the whole time. But you're welcome to join us to help develop those new forms, and we'll be sharing more on the timeline, refined timelines for those in the coming months.
Ana Capuano, Rush ADRC. Thanks for the amazing work. It's amazing to see how much was done. A question that we have is, when uploading the data, sometimes there are like errors that come up, right? Some we think they are proper, but some, that's the real data. That's what we have, right? It's from the participant. So I just wanted thoughts on how this is changing or keeping for the new version. Thank you much.
Do you want me to take? Yeah, thank you, Anna, for that question. So for this version of the UDS, we have done a little bit more work to make the quality checks open, and so there are data dictionaries, and they're available also in programmatic form if you would rather use those. And so having those rules, laid out and both in a human readable and a computable format, hopefully will make it a little bit easier. And the quality rules are based on the actual form design by the clinical task force. And so these rules have been encoded also in the REDCap, and so they're available. But I think to answer your question, they're available also in human readable form. So you can browse them and get an idea of like why you're getting an error instead of like a mysterious like, oh, getting error.
I'll add to just from the data platform side of that. These error checks have been all updated, so they're all updated with the most current information, but also they're separated out into errors and alerts on the new data platform, and right within the new data platform, you're able to clear alerts from within the submission system, so that if it's, checking, did you really mean to do this, because it looks like it could be a mistake, you can clear that, without having to re-upload. So that might.
All right, if we don't have any more questions, maybe just share a little bit more about where people can ask questions at the booths during the meeting.
We have some booths. You should come visit. It's not hard to find them. There's only like 3 booths, so come on over and say hi. Yeah, there's going to be folks from the NAC team there and that are available to answer your questions. If they don't know the answer, they know how to find somebody who does have the answer. So come say hi. Does anyone who's part of the NAC team want to raise their hand so folks can see who some of you are? Okay. I got a bunch of folks here. Yeah.
One booth in particular I would like to plug is going to be the Data Front Door booth that Ben Keller is going to be taking point on. I don't think Ben is here right now. I highly encourage all the data managers to take some time to familiarize themselves with this. You kind of got a primer through that. but this is completely absorbing the current NAC portal at this stage, and so we're going to want to be intimately familiar with it. Those of you who have spent any time with Flywheel, because I know there are a couple of ADRCs out there who have bought into Flywheel as a management platform, will be very familiar with its layout and everything, but Ben and his team have done a tremendous job of working through a lot of the hiccups they had early on, and it's a very, very smooth process at this point, but it does require a little bit of onboarding, so this is a good time to take advantage of that.
Yeah, and Ben will be joining us online for the conference, but Shilpa, who is our tech team lead manager, is here. I'm so glad to have her here. She's a new member of our team, is doing amazing work, and she would be a great person to connect with if you have questions about Flywheel in particular.
Yes, and Ben will be hosting some virtual booths during the lunch hour each day. You can find those times on our high-level agenda. But without further ado, we have one more question. It looks like that just came in online. Does current UDS-3 participants receive a new NAC ID when they complete their initial UDS-V4 visits?
No. You only assign new NAC IDs to newly consented participants at your center. So that's what that system is for. The same NAC ID, if your participants have a NAC ID, that NAC ID stays with them forever. It's going to accompany every single piece of data that comes in for that participant from here forward.
Okay, I think that's all.